Regulation of gene transcription by growth factors and cytokines. Growth factors and cytokines are essential signaling molecules that regulate a vast array of cellular processes, including proliferation, differentiation, survival, and inflammation. They exert their effects primarily by modulating gene expression. This involves a complex interplay of signaling pathways, transcription factors, and epigenetic mechanisms. Overview of the process. Binding to receptors. Growth factors and cytokines bind to specific receptors on the cell surface, triggering intracellular signaling cascades. Signal transduction. Activated receptors initiate signaling pathways, often involving tyrosine kinases, e.g., receptor tyrosine kinases (RTKs), or Janus kinases (JAKs). Transcription factor activation. Signaling pathways converge on transcription factors, leading to their activation or inhibition. DNA binding and gene regulation. Activated transcription factors bind to specific DNA sequences, enhancers or promoters, to regulate gene expression. Key signaling pathways. RTK-MAPK pathway. Activated by growth factors, this pathway leads to the activation of transcription factors like ERK, JNK, and P38, which regulate gene expression involved in cell proliferation, differentiation, and survival. JAK-STAT pathway. Primarily activated by cytokines, this pathway involves the phosphorylation of STAT proteins, which then translocate to the nucleus and act as transcription factors. PI3K AKT pathway. Activated by various growth factors and cytokines, this pathway promotes cell survival, growth, and metabolism through the activation of transcription factors like FOXO and nitrogen monofluoride kappa B. Transcription factors. STATs, signal transducers and activators of transcription. Activated by cytokine receptors, STATs regulate gene expression involved in immune response, inflammation, and cell growth. NF-kappa B, nuclear factor kappa B. Activated by various stimuli, including cytokines, NF-kappa B regulates gene expression involved in inflammation, immune response, and cell survival. AP1, activator protein 1. Composed of Jun and Phos proteins, AP1 is activated by various signaling pathways and regulates gene expression involved in cell proliferation, differentiation, and apoptosis. Epigenetic regulation. Epigenetic modifications, such as DNA methylation and histone acetylation, play a crucial role in regulating gene expression in response to growth factors and cytokines. These modifications can alter chromatin accessibility and affect the binding of transcription factors to DNA. Role in cellular processes. Cell proliferation. Growth factors stimulate cell proliferation by activating genes involved in cell cycle progression and DNA synthesis. Differentiation. Growth factors and cytokines induce cell differentiation by activating genes specific to different cell types. Survival. Growth factors promote cell survival by inhibiting apoptosis and activating genes involved in cell survival pathways. Inflammation. Cytokines play a central role in inflammation by regulating the expression of genes involved in immune response and inflammatory processes. Implications for disease. Dysregulation of growth factor and cytokine signaling is implicated in various diseases, including cancer, autoimmune diseases, and inflammatory disorders. Understanding the molecular mechanisms underlying these processes is crucial for developing targeted therapies.